Good afternoon, and thank you for joining our sessions this afternoon, the ALS Genetics Options Today and Opportunities for Tomorrow. I'm Bob Twasutin from the ALS United, and our speaker today would be Ellie Harrington. She's a genetic counselor that comes out of the Eleanor and Luke Gehrig ALS Center at Columbia University. She offers support to our patients and families and those seeking genetic services. Welcome, Ellie, and thank you for coming.
Thanks, Bob. Thanks, everybody, for being here today. I'll go ahead and share my screen. I'll have a presentation, hopefully for about 30 minutes, and then I'm happy to take questions. Just add those to the chat, and we can leave some time for questions at the end. So as Bob said, I'm a genetic counselor. I specialize in neurogenetics and work at the ALS Center at Columbia. So really my main experience and my main focus is in ALS genetics. So this, just to preface the conversation today, some of the bigger headlines in genetics and genetic testing in this last year was the FDA approval of Qalsody, which is a SOD1 gene-targeted therapeutic. So I'll mention that a little bit today, as well as consensus guidelines for genetic testing for individuals with ALS. So we'll get into those details. Just some of the things we'll cover in our conversation today. I'll give a brief overview of general genetics and ALS genetics, how testing is performed, then questions you all might have. So what's your genetic risk? What's the risk for family members? What are the considerations when thinking about genetic testing? And some of those really include the ongoing trials and therapeutics. Okay, so starting us off with genetic review. For many people, the last time they thought about genetics was high school biology. So I'm just going to provide a very brief overview. I promise I won't get into a Punnett square with you. So we know humans are really complex beings. We're made up of trillions of cells, and within all of our cells contain our complete genetic code. These genes, over 20,000 genes, are packaged onto what we call chromosomes, and each of our genes have a really specific function. So they code for a protein, which is responsible for a function in the body. So some genes code for hair color, some genes protect us against cancer, for example. So when we talk about genetic testing for ALS, we're really talking about testing those specific genes. And you can think about genetic testing kind of like reading through a gene with a spell checker, looking for a spelling mistake or a mutation. And that mutation would cause the protein not to work properly and the typical function of the gene not to function correctly. So continuing with basic genetics, we'll cover inheritance, how our genes are shared within a family. So this is an individual here on our chromosomes. The gray hold our genes. So example of the genes in the light blue here. All of us have 2 copies of each of our genes. And we inherit one from our mother and one from our father. So when you're thinking about perhaps yourself and your children, again, we have you here and your partner, and you pass down either one copy of your gene or the other, and same with your partner. So your children are a 50-50 makeup of each you and your partner. So most genes associated with ALS are dominant in inheritance. So here we have an example of a mutation in one of your 2 gene copies. One mutation is enough to cause disease. And that inheritance is true. If you're found to have a gene mutation, you have 1 gene mutation, 1 normal copy, there's a 50% chance the mutation is passed down, an equal 50% chance the normal working gene copy is passed down. So there's a 50% chance that first-degree family members might also have the same gene mutation. So how does this all relate to ALS and ALS genetics? Over 30 genes have been associated with ALS. And when we break this down, we can kind of work backwards. So there's lots of different pathways, proteins, and cellular functions that keep our motor neurons healthy and functioning properly. So I've listed some of those functionalities here on the right, and genes that code for those functions here on the left. So C9orf72, SOD1, FUS, for example. So when we have a mutation in a gene, it results in a dysfunctional protein. These typical functions aren't working, which leads to motor neuron degeneration. This is just a figure of ALS gene discovery over time. So we have the specific genes and our timeline here on this axis. And the size of the circle, so SOD1 was discovered in the 1990s. The size of the circle represents the contribution to disease. So you'll see the bigger circles, C9 and SOD1, are the most common genes associated with ALS. So I'll provide a few quick details on these two genes since they are the most common. First, SOD1. So the figure here at the top is just that genetic contribution to disease. So C9 makes up the majority of individuals, and SOD1 being the second most common genetic cause. This has autosomal dominant inheritance. And when we think about, as I mentioned, reading through the gene like with a spell checker, we can think this is our huge gene here, and an individual can have a mutation at any different location along this gene. So two common mutations that we'll talk about later in the presentation are A5V, which occurs at the beginning of the gene. And then down here at position 114, there's a mutation called I114T. So two common mutations, but these act really differently and result in very different diseases, as I'll explain. And so one thing just about this is that it's really important to know if you do test positive for a gene, right, there's 30 plus different genes. So not all genetic forms of ALS are the same. And then even if you have a certain genetic form of ALS, each of those is also not the same, since these are really different diseases. So C9 here, this is the most common genetic cause for both ALS and a certain form of dementia called frontotemporal dementia. And this was discovered later in 2011. It's a different type of mutation. Instead of a single nucleotide change, it's called a repeat expansion. I'm not going to go into the details here, but I mainly mention it because it's the type of testing that's ordered is done on a different platform. And so if you've had genetic testing, say at a different doctor a few years ago, one thing you'll want to double check is, was this C9 gene included on the large panel of genes that might have been tested? Because sometimes it can be left off since it needs a different type of technology to test for this specific mutation. So when we're doing genetic testing, one of the incidental findings that we can come back with is a risk for FTD. If you have ALS, you get genetic testing. We find that you have a mutation in this C9 gene. It would help us explain that this is the cause for your disease, but we might also then learn you're at an increased risk for developing FTD in your lifetime. So back to this beginning screenshot of evidence-based consensus guidelines for ALS genetic testing and counseling. This was a paper published by Dr. Matthew Harms, and I, as well as many other individuals, were on this genetic guidelines expert panel, which really concluded that all people with ALS should be offered genetic testing, with a panel that includes at least these genes, the most common ones, and any gene that has an FTD or FDA-approved targeted therapy. So this was only published in 2023, but really important guidelines. Many of the doctors at big academic centers very typically order genetic testing, but it doesn't happen across the board. And so now this has been put into standard of care and guidelines for all people to be offered genetic testing. Quickly a point on how testing is performed. The actual testing process is super simple. So either at your clinic appointment, we can collect a blood tube. We can also send a saliva collection kit to your home to collect DNA. They'll isolate your DNA from your sample, either blood or saliva or cheek swab. That DNA is run through the sequencing technology. And then the information from that is analyzed and we get our test results. So the results from that you can get back on your report are a positive result. We found a mutation. We think it's causing your disease symptoms. A negative result, meaning no abnormalities were found, or an uncertain result. So an uncertain result is abnormality in that DNA spelling of your spell check. We just don't have enough evidence to say, does that cause the gene not to function properly? Is it really harmful and causing disease? Or is it just a benign spelling error and doesn't cause the gene not to function? So those uncertain results are fairly common. Usually we're able to downgrade them and figure out that they're benign and unrelated. Okay, so now we have a background on genetics and ALS genetics. And I want to relate this specifically to you or your family and what questions you might be having. So when we think about your specific risk, the first thing we think about is your family history. That really helps us understand what genetic results we might get back when we send genetic testing. So this is a pedigree. This is a family history. The squares represent males, the circles represent females. If there's a slash through the shape, that means the individual has passed away. And then oftentimes there'll be shading to represent certain diseases in a family. Okay. So here we have an individual with ALS. They have a sister, a son, and a daughter, for example. This is a good example of somebody with no known family history. So first degree, second degree, third degree, all individuals with no neurological disease. And this really makes up the majority of people with ALS are not known to have a family history. And when we run genetic testing on people with no family history, we usually don't identify an underlying gene mutation. So only about 5 to 10% of people with no family history test positive for an underlying causal mutation. So what's the cause of disease if we run genetic testing and don't identify anything? That's the case with most people, right? It can be really frustrating. It's likely a multitude of different small environmental factors and different small genetic factors in combination that over time in one's lifetime, they go on to develop disease. And I'll touch on that a little bit more in another slide. So here's an example of a family history or familial ALS, an individual with ALS, their mother had ALS, maternal uncle with ALS, and maternal grandmother with ALS. So C9, as I mentioned, can also cause frontotemporal dementia. So given that there's dementia in the family, I might be suspicious that the mutation that's causing disease in this family could be C9. So for those with a family history of disease, that makes up about 10 to 15% of people of ALS. We can typically identify a genetic cause in the majority. Roughly 2/3 of people will have a mutation found on testing. So to summarize that, the question is, do you have a family history of ALS? I also ask about related conditions like dementia, Parkinsonism, psychiatric conditions. If the answer is yes, there's familial, we do have an increased risk of it being genetic, finding a gene mutation on testing. If the answer is no, somebody has singleton disease, we're much less likely to have a genetic form of disease. We're much more likely to get negative genetic testing results here. So how does this relate to your family? If you have no family history of ALS, you get genetic testing and your test results are normal or negative, That means you have 2 normal gene copies on all of your ALS-associated genes. You either pass down a normal gene copy or a normal gene copy. So your children, your siblings are not at an increased risk for developing ALS themselves. If you have an abnormal or positive genetic test result, that means you have a mutation on one of your 2 gene copies that carries that dominant inheritance risk. There's a 50% chance for each child or each sibling that they might also carry the gene mutation. So the big question here is if you test positive, say your child gets tested and they test positive, well, what's the chance that they might actually develop symptoms in their lifetime? Okay. So this is something called penetrance, the likelihood that someone will develop disease associated with a certain gene mutation. So when a mutation is 100% penetrant, that means 100% of people will develop disease in their lifetime. When it's 80% penetrant, 80% of people will develop that disease if they have that gene mutation. So I'll walk through a few examples here on the next couple of slides. And I like to use this jar example to really explain that not all risk is equal, and all different genetic forms of ALS are really different from each other. So here we have this jar. We're all at a baseline population risk, so the jar is filled a little bit. And when this jar is filled to this red line, that's when somebody developed symptoms of ALS. And to get to that line, there's different factors. So there's environmental factors that can contribute to the onset of disease, but also different genetic factors that can contribute to the onset of disease. And so people in their lifetime, maybe those without ALS, might have some of those, but it's not enough to get them to the point where they're actually developing ALS themselves. When we're talking about sporadic ALS, so that's 85–90% of people, it's really a combination of small genetic factors and environmental factors that likely contribute to the onset and development of disease. Now, these small yellow circles, these small genetic factors, are not those big gene mutations that we've been talking about in the previous slides. These are really not well yet understood or discovered. That might be really common in the population, but in this specific combination here with the environmental factors, it causes ALS in this circumstance. So these smaller genetic factors are certainly something that that is being studied in a variety of different research studies. But we'll talk about how this also relates to some of the genetic forms of ALS. So here, for an example, we'll list an SOD1 variant. I think I mentioned there's one called A5V at the beginning of the SOD1 gene. And this is a highly penetrant gene mutation. So this genetic factor is really big. At birth, we know that they are at a very increased risk for developing ALS in their lifetime. They're almost at that symptomatic line, and environmental factors, perhaps other small genetic factors, contribute over time to their eventual development of disease. So this is a highly penetrant mutation. Almost everybody with the mutation eventually develops disease in their lifetime. So that's very different from the I114T variant or mutation, which has some reduced penetrance. And we can think about that as just a smaller genetic factor. So they're born with certainly an increased risk, maybe above the sporadic population and above that general baseline population, but not as much as those individuals with an A5V variant. So this is just a good example of the variable penetrance across different genes and even within mutations within the same gene. And that this slide just kind of summarizes that. So here's kind of an example of that within a family. We have an individual who tested positive for a C9 gene mutation. There's a 50% chance it was passed down to each child. And the penetrance here is not 100%. So this pedigree on the right shows the individual with ALS, but they were found to have inherited that mutation from mom who doesn't have ALS. So it's an example of how mutations can be inherited in the family, even in cases where there's no family history of ALS. And so that's really one of the main reasons why genetic testing is offered to everybody, not just those with a family history. Because of this reduced penetrance, these mutations can kind of hide within a family. So considerations for testing. We've talked about a number of these. So some people undergo testing because they are just curious. What's the cause of their disease? I know it's a frustrating process, a long process for many to get to a diagnosis, and genetic testing can sometimes determine if there is a specific cause. On the other hand, if it was negative, testing, might not have been so useful, and we're kind of left where we were before we got the negative result. Depending on the genetic result, it might help refine the disease prognosis. So that I114T SOD variant is a much more slowly progressive disease than the A5V is a much more quickly progressive disease. So sometimes the genetic result can give us some more information about what the disease might look like moving forward. Other times, there are some genes that have high variability across the mutation and even within the same family. So there's some circumstances, of course, where this won't be helpful. Some people want to clarify risks to family members. If there is a mutation in the family, they want to make sure that information is clear and those risks are discussed with family members. But of course, this can also invoke a lot of anxiety. And I know people with ALS already have so much on their plate that the communication of this information certainly doesn't feel like a priority. And so we really have to weigh everything that's going on with you and your family and what makes most sense for you in learning this information. So other considerations, which I'll get into next, are gene-targeted therapies, one that's FDA approved, and kind of the information that we can get from this that can inform enrollment in clinical trials. The opposite side of this is that sometimes, if negative, or if in a gene that doesn't have any clinical trial availability, it really might not change our care plan, or if there is a trial, of course, those also have some risks. So really kind of documenting some of the decisions and thoughts that people have when weighing the options for them going forward with genetic testing. Okay. So I mentioned this at the start of the trial or at the start of the conversation, and perhaps this has already been brought up in a previous talk as part of this conference. But Qalsody was FDA approved this last year, which is a gene-targeted therapeutic. It's called an antisense oligonucleotide. I'll describe that in a few slides, to target the SOD1 gene. So many people are deciding, okay, I want to make sure I have all of the boxes checked for potential treatment options. So to be eligible to receive this therapy, of course, individuals need to have a mutation within the SOD1 gene. In order to learn that, they have to go forward with genetic testing. So this is kind of a huge success in the genetics and gene-targeted therapy world of things. One thing that I want to relate back to families is, okay, well, if I test positive, maybe someone doesn't want to enroll in that, someone doesn't want to receive the Qalsody drug themselves. But we can also think about how this might impact other family members. So that same drug, Qalsody, is in clinical trial for asymptomatic family members who got tested and carried a specific gene mutation. So if you test positive for SOD1A5V, if maybe a sister or a sibling or an adult child gets tested and they're found to have that same mutation, they might be eligible for this clinical trial, which is treating individuals pre-symptomatically before the onset of disease symptoms, really hoping to prevent disease from occurring or pushing back the onset of disease. So one thing I want to mention here is just taking a step back and thinking about the bigger picture of things. This Qalsody was FDA approved this last year, and this ATLAS study started about 1 or 2 years ago. But 5 years ago, this wasn't FDA approved. There was issues with the clinical trial. And so even within this last 5 years, there's been a lot of success. And so I think when you're thinking about the option for you to get genetic testing, we also need to think about the future. So in 5 years, might there be therapeutics targeted for a gene that might have been identified for you? And could that really have some huge implications for preventative treatment for family members down the line? And so this example of this pre-symptomatic trial, I think, is really important to bring to the bigger picture of other genes and what might happen for that moving forward. So just to clarify a few things, I want to talk about some of these clinical trials, these gene-targeted clinical trials. And much of these, as I say, gene-targeted, the drugs are really, for the most part, antisense oligonucleotides, ASO therapies. So we know our basic genetics, our DNA codes for our messenger RNA, which creates that normal working protein. When we have mutated DNA, that is translated to mutated mRNA, and we have a disease-causing protein instead. So an ASO therapy basically acts like a tag for latching onto the mutated mRNA and puts a big stop signal on that mRNA, so it can't create the abnormal disease-causing protein. So these types of drugs have been successfully developed for other genetic diseases. The most noteworthy are spinal muscular atrophy and infantile motor neuron disease, Duchenne muscular dystrophy, FDA approved with high success. And so the Qalsody drug is another example of the first ASO approved for ALS. So we mentioned those. SOD1, Qalsody went through long clinical trial before it was approved, and now the ATLAS is active for pre-symptomatic individuals. Individuals with FUS mutations, there's also an active ASO clinical trial called Fusion. There's another ASO for ataxin-2. This is available for people both with ataxin-2 mutations and individuals without ataxin-2 repeat expansions. So a little bit different of a drug trial there. And then the C9 related ASO was discontinued last year as it failed to show clinical benefit, and I think there was some adverse side effects. So last few slides here, what about the other genes? What about, the ones besides C9 and FUS, these others that are at more, occur at smaller frequencies? So at Columbia, we launched a program in the last year called Silence ALS. And our goal with this program is to develop these types of ASOs for those more rare ALS-associated mutations. So we're focusing on a few right now: TAR-DBP, ANXA11, CHCHD10. So we work with, we have a partnership with the N. Lorem Foundation, whose main goal is to develop ASO drugs for rare genetic diseases. And so we've had success in developing 2 and have given the drugs for 2 individuals, one with a TAR-DBP mutation and one with a CHCHD10 mutation. So hopefully more of this to come with many more people in the pipeline for this project. So the last 2 points that people often ask questions about are family planning options, either for themselves or for their children, and potential insurance discrimination risks. So first, touching on family planning, if you are tested, you're found to have a gene mutation, and say your daughter is tested, she's also gene positive. We can now use the process of in vitro fertilization and embryo screening to ensure that if she wants to have children of her own, perhaps she wants to ensure that that family mutation is not passed down to the next generation. So a very brief summary of in vitro fertilization: stimulating a woman with hormones to overproduce the number of eggs in a cycle, retrieving those eggs, fertilizing those eggs with sperm to create an embryo, growing the embryo, and then reimplanting an embryo into a woman to produce a pregnancy. So that's pretty standard IVF process that's been happening for years and years, beginning mainly with people with infertility issues. So there's now the process of pre-implantation genetic testing for mutation or monogenic diseases. So before an embryo is implanted, they can now test those embryos, screen which ones have a mutation, which ones don't, and only implant an embryo that doesn't carry a gene mutation. So for those individuals who, have SOD1A5V, it's a highly penetrant mutation. It's affected, 10 family members. Some individuals are really interested in going through this process and making sure, if they want to have biological children of their own, that perhaps they can stop it from occurring in the next generation. Okay. Lastly, it's always a big question: potential discrimination risks. And I think I put this in italics in my last slide because really it's a potential, and it's really something that I say we cross that bridge if and when we have to come to it. So GINA is a law that protects all of us against insurance discrimination from our health insurance companies and our employers if we work at a company that's more than 15 people. So for example, if I have a cancer gene mutation, I'm at risk for developing cancer in the future. My health insurance still has to cover all my healthcare needs. My employers, Columbia, can't fire me because they know I might develop cancer in the future. GINA does not protect against life insurance, long-term care insurance, or disability insurance. Okay. So for you, if you're somebody who has ALS, this is kind of a moot point. If you're applying to life insurance right now, they will likely discriminate against you based on what's in your medical record, and a genetic result doesn't really add much to that. So discrimination risks for an individual who has symptoms that are documented in the medical record, moot point. This is really, we think about when we're testing those asymptomatic family members. So if you test positive, you have a causal gene mutation, and if your relatives want to get tested, we just make sure that they're properly insured before they themselves go through genetic testing. So cross that bridge if and when we have to come to it. So I think that's kind of covered a lot of the considerations for testing. I'm sure there's some that you all think about as well, or maybe this has prompted some other questions and ideas, considerations. So I'll go ahead and stop now, and I'm happy to take questions.
One question come through the audience is C9 caused of so many ALS cases? There doesn't seem to be many clinical trials. How likely will it be a cure or a drug within the next 20 years?
I know, good question. So it is the most common genetic cause. And I'll put into perspective that it was only discovered in 2011. So we are, further out from SOD1, for example, which was discovered in the 1990s. But we've also had a lot more success in the last 5 to 10 years of gene-targeted therapeutics. And so I don't want to make any promises, but I feel and I hope and I believe that there will be much more success for C9 targeted therapies in the next 10 years. Yeah.
And another member of our audience asks, are there any preventative strategies for those who have familial history of ALS?
The quick answer is no. For SOD1, there's the clinical trial, and we're researching some of those environmental factors that might, contribute to the onset of disease. But it's so different in different populations that those environmental factors are difficult to specifically determine. So we're actively working on that, but there's no great answer to that question.
Another member asks, my maternal uncle and my father both have ALS. What are the chances that it is genetic? What is the probability that myself, my brothers, and my children will develop ALS?
Yeah. So we consider a strong family history of ALS to be 2 or more individuals. So I'd say, that sounds like it's familial. There's more than one person, 2+ people that might have ALS, and it's more likely to be a genetic cause of ALS. So if those people undergo genetic testing, I'd say we'll likely find a gene mutation. If that's the case, there's a 50% chance that you or your siblings might also carry the same gene mutation.
Okay. And yet another person asks, for those with a familial history of ALS C9, what are the pros and cons of genetic testing for asymptomatic individuals? If the gene is present, does it always mean that the ALS will manifest, or some trigger needed for ALS?
Pros and cons of testing are really individual, and it really depends on where you are in your life stage as well. So that family planning might be an option and a consideration for people who are younger. Some people are retiring and they're thinking about moving, so maybe they would make financial planning decisions. They'd buy a 1-story house instead of a 2-story house. So people have many different considerations when going into testing. I think the most important thing when making that decision is knowing why you're getting tested, why you want to learn this information, how it will impact some decisions you might be making. And those motivations are really important and really impact the outcome, psychological outcome of how people do once learning that type of information. I think there was a second part to that question, and I think it was regarding penetrance. So if you were found to have the mutation, will you necessarily develop ALS? The quick answer to that is no. C9 is not 100% penetrant, and the penetrance is variable. So I quote around 80%, but it really could be anywhere between 50 and 85%, I would say. And so that's a big consideration too, is if you get tested, we could tell you have the mutation or you don't have the mutation. If you do have the mutation, we won't be able to say, well, are you going to get disease? What is that going to look like, ALS or FTD? When might that occur? How might it progress? Those are questions that we don't have answers to right now. So when thinking about testing, those are some of the important things to think about, is how you might process and cope with those remaining uncertainties that might exist.
Another audience member asked the question, I had genetic testing done at Columbia in 2021. Was C9 included during that time?
Yes, I'm confident it was. If you want to double check with me, feel free to send me an email though, and I can make sure to send you a copy of those results.
We have another couple of questions. If C9 is the cause of so many cases of ALS, there doesn't seem to be many clinical trials. How likely will there be? I think we got that earlier. Also, I was told that children with someone with C9 will not necessarily develop ALS at an early age, but then read another study that it would come out earlier. Is there any additional information on that?
Yeah, This is, I would say the quick answer is that children of someone with C9, it's not been proven that it occurs at an earlier age. This has to do with the idea with that type of mutation, a repeat expansion. Many other repeat expansions conditions, the size of the repeat correlates with disease onset and disease severity. And when you pass down a repeat, it can increase in size. But that size of the repeat for C9 doesn't relate to disease onset, the age of onset. And so even if it does expand and it's a bigger size in the next generation, it doesn't necessarily cause it at an earlier age than in a parent. And we see that in many, many families, you know, where that one example of the pedigree I gave, where someone had C9 ALS at 55 maybe, and their parent had the mutation and didn't develop ALS at all. They had a child who also didn't develop ALS. So no, not necessarily.
And yet another member of our panel asks, my husband has C9 and his dad passed from it. He's from Finland, one of our highest diagnosis rate globally. Are you working with other countries for research?
Yeah, we collaborate with researchers, especially kind of for the Silence ALS program and for the ALS Families Project, which is specific for asymptomatic individuals at risk for inheriting a mutation. So we have participants from all over and certainly collaborate with other researchers.
And does insurance, most insurance covers genetic testing, or are there some carriers that have excluded it?
Good question. And I took this slide out because there are at least two, maybe more, sponsored genetic testing programs, one at Prevention Genetics, one at Invitae. And so when I order somebody genetic testing, I don't even enter insurance information, and it's completely covered, free of charge. So shouldn't be an issue, at least for now. These usually last a few years. So I used to have costs on one of those considerations in the weighing of tables, and really cost is not a consideration anymore.
Right. And when you have genetic testing done, how long does it usually take for the results to come back prior to getting those results?
Yeah, good question. Depends which laboratory we send to, but after the sample reaches the lab, anywhere from 3 to 5 weeks.
Very good. And you were mentioning in the last slide about Gina, just for the audience knowledge, one of ALS United's advocacy reach out efforts in the last month was speaking to New York State legislators about trying to get that coverage for identity for those ones that are non-exclusive at this time. So the disability, the long-term care, and the life insurance, because some families are hesitant to jeopardize coverages. So there is an outreach by many, as well as the ALS United, to try to get that covered under the legislation at this time, especially for New York State. I'm sure other states are trying to cover the same. Are there any other questions? I have gone through what the chat box has had at this time. We're still open for some time if you'd like to present a question for Ellie. Just when people are pursuing genetic testing, what does it involve? Is there questionnaires? Are there testings? Are there serums involved?
I think I had a slide on this, so it's pretty simple. It's just providing a tube of blood, a saliva sample, a cheek swab sample. Hopefully you'll have genetic counseling and you'll sign a consent form that approves your sample will be used to test for the known ALS associated genes. Then it gets shipped to the lab performs the testing, sends us the result, and we'll share that result with you.
Very good. Once again, I open for additional questions if there are any. Let me just check questions and answers for the last. I think we have everybody covered under that section. I don't think we have any more questions. Ellie, any closing remarks on?
No, I think, genetic testing is, why I titled the talk, considerations for now and the future is, this is an evolving field. And we're learning more and more about the genetic contribution to disease. More and more therapeutics are being coming into clinical trial and really in the preclinical trial phases. So before they're offered to people with, humans with disease symptoms. And so even if testing might not be applicable now, it might be for the future, whether that's for you or whether that's for a family member. The last thing I didn't mention, and I should have in a slide, is the majority of people have sporadic disease. Their testing comes back negative. And so as we think about how that impacts family members, sometimes a negative result can be really impactful as well. It can reassure family members that there's not an increased risk for them. And I think flipping the script a little bit and thinking about your actual risks based on your family history, based on our knowledge of how many people actually test positive, we can think about, well, what are the benefits of getting a negative result back as well? So that really helps us, one, think about family and their risk in a different way, and two, really narrow, if you're someone who's interested in clinical trials, it narrows us to look at those that are offered to everyone, regardless of if they have a gene mutation, and cross out those ones that are gene targeted. So it can help focus our clinical trial search as well.
Very good. Thank you, Ellie, for an informative session, very highlighting and very easy to understand. I want to thank everyone for joining us again. And please join us for our next session at 4:30 p.m. Navigating ALS with Children and Youth. How and when to talk about ALS. Our speaker will be Melinda Kavanaugh. We welcome you to the next session again at 4:30. Thank you for coming.
Thank you.